This is in Cologne. Before he was in Essen, and it's there that I met him for the first time around 20 years ago. And he has been invited here in Florence in the context of an internationalization process for the PhD. So the university, supports the invitation of person to give PhD courses, and so he has passed the last few weeks here to give a course on the statistical physics of biological evolution. And so, before leaving, he's going to leave on Saturday. We asked him also to give a general talk within the series of Sguardi, sulla complessità, views on complexity. And so now he's going to speak about how predictable is evolution. Thank you.
Okay, thank you very much, Paolo. I'd like to thank Paolo and Roberto for inviting me here and giving me the opportunity to spend a few weeks in this wonderful city, and also to give a seminar here today, which is sort of revolving about this question of predictability of biological evolution, and I will be giving you an opportunity to present some of the work that we have been doing in Cologne in the framework of these two collaborative research centers funded by DFG. So if we look at the biosphere, then of course the first thing that strikes you is an enormous diversity. Right? So there's a mind-boggling diversity of life forms, but at the same time you can also observe many instances of what is called parallel evolution, where unrelated organisms have, mainly through adaptation, have evolved very similar shapes and very similar functions. And one proponent of the view of that these constraints are very important, that therefore life in some sense is much less diverse than it could be, is Simon Conway-Morris. And in his book you can find many examples like this. This is one of my favorites. This is the evolution of saber-toothed tigers in the placental mammals like us and in the marsupial mammals, which are animals like kangaroos. So you see they look very similar, although they are not strictly speaking related. As humans, as a species, for the species of humans, it's probably a good thing that both of them have now gone extinct. But nevertheless, these and many other examples provide food for thought about how determined evolution actually is. And of course, since Darwin, we basically know, at least roughly know, the mechanisms that lie behind the evolutionary process, and we know that it is indeed an interplay between deterministic and stochastic forces, deterministic being selection that tries to adapt organisms towards a certain function, and stochastic effects that come from the reproductive events, the randomness inherent in leaving offspring, and also mutations. And so Stephen Jay Gould, the great evolutionary biologist from the United States, phrased this question in this way a couple of years ago. So he asked if we could replay the tape of life, that is, if we could start life again on a planet similar to the Earth, would the outcome be similar to what we have, or would it be completely different? Gould had the view that it would probably be completely different. He believed that contingencies and random events in evolution are very important, whereas Conway-Morris has written many articles and books where, for example, he claims that if we one day would meet intelligent beings in outer space, they would probably look like us because things are so much constrained. So this is a debate that has been going on in biology for at least 100 years. And of course, it's a very difficult question, and there is no chance at the moment to really replay the table of life, although with the discovery of extrasolar planets, maybe eventually we'll have a chance to see how things have developed elsewhere. However, there is a field of research called experimental evolution, which is now able to address these questions in a more systematic way. So these are experiments that are conducted with microbes, typically with bacteria or viruses, and that can propagate these populations in parallel over long times, and they therefore address predictability in the weak sense. So we're not talking about here predicting evolution the way we predict the weather, to say what will life look like 1 million years from now, but we're using predictability in a weaker sense where we're asking how repeatable are adaptive trajectories. That is, if I have 100 bacteria colonies evolving in similar conditions, how different will they be after a certain number of generations? So these are experiments that have been conducted over the last decades and are being done in many laboratories, and they provide much inspiration for the kind of work that we're doing here in theory. So the first thing that you notice in these experiments is that the fitness of these populations increases. Now, how do you measure fitness? So the term fitness will occur in this talk many times, and essentially fitness is a measure of reproductive ability. Think about fitness as the mean number of offspring that an organism leaves in the next generation, and this can be monitored in experiments by competing the evolved organism against the ancestor and seeing how much quicker they reproduce. So I will give you more examples of these kinds of experiments as we go along, but just as a kind of introduction, I wanted to show this slide, which is from the classic evolution experiment, which was started by Richard Lenski in 1988, with 12 populations of E. coli in a glucose-limited medium. That is, in a medium where these bacteria could grow, but they didn't grow very well. So they had sort of space for improving. And this is now from a paper a few years ago. At that time, this experiment had been conducted for 50,000 generations. And here you see this fitness increase. So this is the fitness relative to the ancestor, which has increased by about 50%. One thing that has happened in some of these populations is that they have evolved so-called hypermutator strains, so they have increased their mutation rate, which in this case also implies that the fitness increases somewhat more quickly. But by and large, on the level of fitness increase, the 12 populations are rather similar. However, if you look at them in more detail in terms of the actual genetic evolution, you see many differences. And so there's an enormous data set that has been come out of this experiment now that is currently being analyzed by various groups to try to quantify how much parallelism there is in these different populations. Now, what I want to do in this talk is to discuss 3 case studies from our own work where different factors that are important for evolutionary predictability could be quantified in a simple way. And we'll go from something very simple to something more complicated. So I'll start off discussing the sort of elementary process, a single step of adaptation. Then we'll talk briefly about mutational pathways where several mutations occur after each other. And finally, I will talk a little bit about the effect of population size on predictability, like with other natural phenomena or even phenomena in society like elections, for example. You expect things to become more predictable in larger populations, and this is something that we will discuss here. So let me start with the first story, which is about a single step of adaptation. So what is a single step of adaptation? So these bacteria populations are asexual, so the only way in which they can produce diversity is by mutation. So the elementary event is sort of schematically illustrated here. So we have the current genotype, so this is sort of the current population, and it has access to a certain number of mutants, right? So there are certain mutations that can occur starting from this initial genotype, and some of them are deleterious, marked in red, and others are beneficial, and some of them will increase the fitness, right? So the elementary event is that one of these mutants appears, and if it has a larger fitness, that is, if the reproduction rate increases, then this mutation can spread in the population, and eventually the entire population will carry that mutation. This is a process that is called fixation. So the step of adaptation means that one of these beneficial mutations will be fixed. And so on this level, the question of predictability simply means what is the probability that if I run 2 replicate populations that the same first step will occur. And this may be useful here to use a kind of analogy. So if my 2 populations are sort of throwing this dice, I can ask what is the probability that 2 dice show the same number of dots. Okay, so if this is a fair dice, I'm sure that you can immediately answer this question. So what is the probability that 2 dice will show the same number of dots?
6 divided by 36.
Yes, exactly. So 1 by 6. Now, of course, here the dice are not really fair because the different mutations have sort of different advantages. So the question is essentially that of having sort of dice that are loaded. Okay, and this is the basis of the simple theory of parallel evolution that was formulated by Alan Bohr a couple of years ago. So we consider a situation where we have n beneficial mutations that are available, and each mutation has some selective advantage. So this is the difference between the reproduction rate of the mutant versus the ancestor with some number Si. Now there's a theory that I have discussed at length in my course that tells you that the probability that the ith mutant goes to fixation when Si is small enough is 2s. Okay? And so the probability that the ith mutant is the first to fix is therefore Si divided by the sum over all the Sj's, which sort of correspond to the other possible beneficial mutations. And the probability that the same mutation is fixed twice is pi squared, which I then have to sum over all the i's, because I don't care about which mutation this is. So this is the probability of parallel evolution in this very simple setting. Okay, and so for the problem of the two dice, this would give you exactly the answer of 1 over 6. So if all the pi's are the same, this would be 1 over n. Okay. Now, in order to calculate this, one needs to know something about these SIs. And for our purposes, we can think about these SIs as being random numbers that are drawn from some distribution. Now, of course, this distribution in general is not known, but there is a hypothesis that was formulated by Gillespie and Orr, which is a so-called extreme value hypothesis, which says that one can use extreme value theory to at least sort of limit the possible shapes of this that this distribution might have. And the argument is that if the organism is viable, then any mutation that makes it even more viable will be in the tail of the fitness distribution, right? Because most of the mutations, of course, will make the organism much less adaptive. And so whatever the adaptation does, it will increase something that is very good to something that is a little bit better. And therefore, one can assume that one is certainly in the tail of some unknown distribution. Now, extreme value theory tells you that these tails have only a limited possibility of shapes. So there are bounded tails, there are so-called Gumbel tails, which are exponential-like, and there are so-called Fléché tails, which have power law behavior. So these are the three classes. And so basically, one can assume that one is in one of these classes and then try to calculate this quantity P2. And what Orr did was to calculate it for the Gumbel class, and he found that it's essentially 2 by n. So instead of having 1 by n, you have 2 by n. So predictability increases in some sense by a factor of 2. However, if this distribution is heavy-tailed, then things become more interesting. So there's a calculation that you can do, basically borrowing ideas from Derrida, from spin class theory, which tells you that for a heavy-tailed distribution, there are sort of two regimes. If the distribution doesn't have a second moment but has a first moment, then this P2 will go, rather than 1 over n, it will go with a smaller power of n. And if alpha is, if this exponent is less than 1, then P2 actually becomes independent of n. So even if you have an infinite number of mutations, the probability that we will see the same mutation twice remains finite. Now, this might seem like a kind of academic problem. So the question is, what are these distributions actually in reality? And it turns out that by now, so there has been a lot of work on this over the last 10 years or so. By now, all three classes have been identified in experiments, and in particular in joint work with Ariane Fisser and collaborators at the University of Wageningen, we have investigated a system which falls into the Fléché class. This is a particular enzyme called beta-lactamase, which confers antibiotic resistance against penicillin. So this is an antibiotic resistance enzyme. This is what the protein looks like. This protein has, so there are about 2,500 possible point mutations that can occur in this protein. And by a kind of exhaustive screen, Mark and Schenk identified 48 of those that increase resistance against a certain non-penicillin antibiotic, which is called cefotaxime. So we have 48 beneficial mutations which we can characterize, and we can then try to evaluate the distribution of effects. And what one finds is that distribution is indeed heavy-tailed, and alpha is approximately 1. Okay? And it seems to decrease with increasing antibiotic concentration. So as you increase antibiotic concentration, the distribution becomes more and more heavy-tailed. So the prediction is that correspondingly the predictability of the first step of adaptation should be very high. And you can evaluate this. So this is this blue line shows the quantity P2 that I showed you. This is sort of the baseline 2 over n plus 1 that you would have if the distribution were not heavy tail. And you see that it sort of increases to about 20% as we increase the antibiotic conservation. And similarly, this quantity, which is a probability that a particular mutation that has the largest effect will fix first, goes up to almost 40%. And this has also been confirmed in a sort of direct experiment using replicative lines, which shows that in 10 out of 12 of these lines, the largest effect mutation is the first one to appear. So the conclusion here is that in this particular system, the probability of the same mutation occurring twice is very high because the distribution of effect sizes is heavy tail. Okay, now of course in many cases, a single mutation is not really enough to produce a significant effect on fitness, so one has to think about what happens when several mutations occur after each other, and this is the problem of mutational pathways. So for this one has to look at these structures which are called hypercubes. So this is an example where we have 3 mutations. So suppose that there are 3 mutations that can take me from this initial type with all zeros to an adaptive type that has a 1. So 0 means the original type, 1 means the mutated type, and this can occur in 3 different places. And this is sort of an illustration where the fitness of these various combinations are illustrated by the sizes of these balls. So this would be the fittest type, and if you want to evolve this 3-fold mutation, you have to perform 1 mutation after the other. Now, because there are 3 mutations, they can occur in 6 different orders. So in general, if there are L mutations, L factorial different orders, and they correspond to different pathways. Right? So each pathway corresponds to a particular order in which these mutations occur. And the interest here is now in those pathways along which adaptation is actually possible, and for that, at least if the rate of mutation is small, for that it is necessary that each step by itself is already conferring an advantage. So each step by itself should increase resistance, okay, or fitness. Now this kind of analysis was first done by Dan Weinreich and collaborators in about 10 years ago. And this is the title of their paper. Darwinian evolution can follow only very few mutational paths to fit a protein. So the context of this work was clearly also the predictability of evolutionary pathways. It's the same system that I discussed just before. It's again this beta-lactamase enzyme, but in this particular case, rather than looking at a large set of initial mutations, they looked at 5 different mutations that in combination have a very large effect. So these numbers that you can see here are measures of antibiotic resistance. It's essentially the concentration of the antibiotic that these bacteria can tolerate, and that increases from 0.09 to 4,100. So there's a big increase in resistance if when these 5 mutations occur in combination. And so what Dan Weinreich did was to construct all the intermediates. So because there are 5 mutations, there are 2 to the 5 or 32 intermediates, and all of those can be constructed genetically. This is not very difficult anymore these days. And then for each of these combinations, you can measure the resistance, and you can ask how many ways are there in which you can combine these 5 mutations. So how many of the 120 in this case mutational pathways are actually uphill? And the finding was that out of those 120, only 18 are actually uphill in this sense. Okay. This is so the context or the relation between accessibility and predictability. As I said, these pathways are accessible if the fitness or resistance increases monotonically. And so Weinreich's interpretation was that the existence of a small but non-zero fraction of such accessible pathways implies at least high retrospective predictability in the following sense. So if I have arrived here, so I have this adaptation, and I would like to retrospectively tell how the system has evolved here, a priori there are 120 possible pathways, but a posteriori, if I know the landscape, I know that only 18 of those are actually possible. And in fact, you see that these arrows here have different thicknesses, so you can also assign a kind of weight to different pathways. And if you do that, then you see that only a handful of pathways are really highly probable. Okay? So the picture of Weinreich was that in general, in these kind of landscapes, there is a small but non-zero fraction of such pathways. Non-zero because if there is no pathway, then you cannot even evolve there, right? So then the goal is sort of inaccessible. Now the question that we asked from a theoretical point of view was whether this is typical. Okay, so should you expect that you have such a small fraction of accessible pathways? And as a kind of null model, you could do the following. You could just say, let's assign the fitness values randomly to genotypes. So this is something that we nowadays call the House of Cards model of fitness landscapes. This was introduced in this context in the 1980s. And then you can actually do some rather simple calculations. For example, if you now want to know what is the probability that a given path is accessible, you just have to think about what this path actually does. So such a path is just a sequence of L random numbers, right? So you have L random numbers. You want to know the probability that these L random numbers are ordered increasingly. Now, of course, since all possible permutations of these random numbers are equally likely, the probability that they are increasing is just one particular permutation is 1 over L factorial. Okay, so the probability that a given path is accessible is L factorial. On the other hand, there are L factorial paths, so the expected number of such paths is 1, okay, which is independent of the size of the system. Now, this seems, on the surface, this looks like a kind of strong confirmation of this idea that there is a small but non-zero fraction of paths that are accessible. But this turns out not to be true. So if you look at this problem in more detail, you find that the mean is in fact not at all representative. And if you look at the distribution of this quantity, you find that in fact most landscapes don't possess a single accessible path. So the typical number of this number of accessible paths is actually 0. On the other hand, if you condition on the existence of such a path, or if you condition on the initial point having a particularly low fitness, then you find that the typical number of paths is in fact of order L, and this was proved mathematically by Sguardi and Martinson. And so in that sense, you have a kind of reduction, so that the total number of paths is L factorial. Typically, you have L of those which are accessible, which in some sense can be seen as a kind of confirmation of the scenario, although of course one must say that this null model of course is not realistic, so one would have to think about how to introduce correlations and so on, and we have done that, but this is not really, I don't really have time to go into that in more detail. So let me finally come to the last point, which is the relation between predictability and population size. So one of the stochastic forces in evolution is what biologists call genetic drift, and this is related to the fact that the population is finite, which simply means that as you go from one generation to the next, you have a kind of random sampling process of individuals, and by this random sampling process, even individuals that have high fitness may not leave any offspring at all. Right? So this is a source of randomness, and you expect that this randomness becomes smaller as the population gets bigger. So a priori, you expect that things will become more predictable in large populations. And again, this is something that has been seen in experiments. So this is an evolution experiment, again, with E. coli from the group of Ariane de Visser, with 2 different population sizes evolved in a rich nutrient medium, which one expects to be representative of a fairly complex fitness landscape. And what you see here are the fitness trajectories in the small and large populations, and you can sort of see with your naked eye that the trajectories are more variable in the small population than in the large population. What you can also see, but I don't really have time to go into that here, is that in the small population there are some trajectories that start out very shallow, but actually reach rather high fitness at the end. So there are sort of 2 different modes of evolution here. But the main point is that, things become more deterministic. And this one can sort of explain theoretically and also quantify. And the basic idea is maybe to go back to this picture that I showed you in the very beginning. So if we have this kind of situation, so here we have assumed that only one mutant arises at a time, and then this mutant will arise with a certain probability, which is given by the fixation probability. Now, as the population gets bigger, it of course produces more and more mutants. So there will be several mutants present at the same time, and then they will compete with each other, and then if several mutants compete, then the largest one will typically win. Right? So large populations tend to lead to what we call greedy dynamics, in the sense that the large fitness increases are favored. Okay, so this is sort of what one should expect. Now we wanted to study this question in a bit more detail, and so what we did was to turn to a particular data set that also comes from an experiment. In this case, it's not bacteria, but this is a so-called black mold, Aspergillus niger, which looks like this. And for this system, our Edificio and collaborators generated a data set that is in some sense L-dimensional. So they looked at 8 different mutations that were all deleterious and that reside on different chromosomes. And because they reside on different chromosomes, it was possible to sort of mix them and to generate again all possible combinations. So here we have again a fitness landscape, but a rather large one, with altogether 256 different combinations. 186 out of those were actually identified, and we believe that the remaining 70 are not viable, so they have fitness 0. And for these combinations, fitness was measured for 2 replicas per strain. And so we have a kind of empirical fitness landscape that is rather large. And on this fitness landscape, we decided to study this issue of population size. And so this is done in the following way. We have a population of size n. These individuals reproduce asexually in discrete generations. This is called the White-Fisher model. So each generation, you just replace the resident population by a new population. You have a certain mutation rate. You have some optimum somewhere, and you start your evolution from the viable genotype at some distance from the optimum. And then what we wanted to do was, to do a kind of virtual evolution experiment where we run many of these trajectories in parallel and follow how they move in the space of genotypes. And we did this in 2 ways. On the one hand, you can track this called lines of descent, which means that at the end of the simulation, you look at the most populated genotype, and then you try to reconstruct where it came from. So for each genotype, you basically reconstruct which was the preceding one from which it differed by single mutations, or you can follow the path of the most populated genotype. But here I'm just going to discuss the lines of descent. And now to quantify the predictability, what we did was to look at the entropy of the distributional pathway. So we do many experiments, many simulations. We count how many times a given pathway occurs, and then we calculate the entropy of that distribution as a function of population size. And so this picture shows what these pathways look like. Of course, it's not so easy to visualize the evolution in this 8-dimensional space. So what you see here is a kind of projection onto the plane. This is the mutational distance from the target, so to speak, from the fitness maximum, which is at 0. So here we start at distance 7. And these are all the genotypes at distance 6, 5, 4, and so on. They are organized here in some way that doesn't really matter. And these arrows show you the steps that have occurred in a certain number of runs, for example, 1,000 runs. And if the arrows are thick, then this means that this step has occurred many times. And the population size, or the mutation rate, is 10 to the minus 5. The population size changes between about 100 and 10 to the 7. And so this is a small population. And here you see that things are very random. Right? So this is very much a random situation. So most of the populations don't get much further than distance 1, but a few of them get further, but this is all very random. Now, as you increase the population size, this graph becomes much more regular. Still, most of the populations don't get closer than distance 6, but here you see that a particular pathway is coming out to be quite dominant, which takes you to distance 4. So here you would say that things definitely become more predictable. However, as you make the population even bigger, here you see that some more diversity comes out again. Okay? And so in fact, if you now look at the entropy, so this is now the quantification of those pictures, this is now the pathway entropy averaged over 46 starting points at distance 4. And this is as a function of n, and this g here is just a way of representing the mutation rate. So the black symbols have a mutation rate of 10 to the minus 5, the purple symbols mutation rate of about 10 to the minus 8. But what you should see here is that each of these plots has a minimum. So the entropy goes down, so the evolution becomes more predictable. But eventually these curves go up again. So there's a minimum, and at large population sizes, things become less predictable again. Now, to see where this comes from, one can scale the population size with the mutation rate, and this is done in these two panels. So you see here, this is now n times mu. So this is essentially the rate at which single mutations occur. Right? So single mutations occur at rate mu. So in the entire population at rate n times mu, and you see that the declining part of the curve collapses if you scale it by n times mu. So this is the effect of the single mutational steps becoming more greedy. But if you scale things by mu squared, then you see that the increasing part collapses. And that tells you that this increasing part has something to do with double mutations. So our interpretation is that entropy varies non-monotonically with population size, because as you increase the population size beyond a certain limit, double mutants start occurring, and so the constraints due to the pathway become less relevant, and things become less predictable again. So I should say that this phenomenon so far has not been observed experimentally. There is an experiment that has looked at 3 different population sizes, but I don't quite see this predicted increase. But I hope that this will eventually be seen in experiments as well. Okay, so let me summarize. So I've discussed here 3 different factors that contribute to evolutionary predictability: the distribution of fitness effects, the interactions, which are called in evolutionary biology epistatic interactions, that determine the structure of the landscape and thereby also the pathways, and the mutation supply, that is the amount of mutations that you generate, that is determined by the population size. And there's a further kind of review paper that we wrote fairly recently about fitness landscapes and predictability, as you can see here. So, of course, I'd like to thank my collaborators, students and postdocs at the University of Cologne, and our experimental collaborator, Fisser. And finally, I would like to leave you with a quotation by one of the pioneers of mathematical population biology, Haldane, who said something that I think applies to many fields of science and also to many fields of complex systems research, where he says, no scientific theory is worth anything unless it enables us to predict something which is actually going on. Until that is done, theories are a mere game of words and not such a good game as poetry. And I think in the city of Dante, this will be appreciated.
Finally, the summary have not given an answer to the question posed in the
Of course, yeah.
So what might be the answer?
You know, like you say, literally, Q&A, I mean, more. Evolution is more or less predictable. No, I mean, I think everybody will realize that this is a very complicated question, and the answers are very partial. Certainly the kind of answers that a theoretical physicist like myself can contribute are very limited. I think it's interesting that people like Conway Morris have very strong opinions about this. So he really believes that that, as I said, that extraterrestrial beings will have 5 fingers and 2 ears and 2 eyes and everything. And he knows a lot of biology, and he knows an enormous amount of biology, much more than I do. So maybe there's something to it, but it's of course also something that is very much debated among biologists. So I cannot really say. What I want to emphasize is that it's a question that is now, at least in the context of these simple microbial systems, that is beginning to become accessible to quantitative analysis. For example, a kind of more practical question, rather than asking about the whole biosphere, a question of very practical importance is the ability to predict the next strain of influenza. Right? So if you want to make an influenza vaccine, you have to know a year ahead what is the dominating strain. And this is a field where physicists are now beginning to contribute, and where sort of more refined models and ideas like the ones that I've presented to you might actually become relevant. Or more generally, the evolution of antibiotic resistance, I think, is a field where quantitative ideas of this kind can become useful.
So, question.
Yeah, so evolution is an interplay of the environment and the organisms. So about the environment, if you change the environment, it will change the pathways.
Of course, yes. I mean, this is of course another level of complication that is sort of left out here. I mean, the nice thing about the experimental evolution context is that there you can keep the environment fixed, or you can change it as you like. I mean, you can at least control it. Of course, that also limits, I think, in the view of many biologists, it also limits what these experiments can actually do, because in nature the environments are never really constant. I mean, predictability of the evolutionary process in a changing environment would have to be given, a certain environmental history. So if you impose an environmental history and you place a population there, then of course its evolution will be influenced by the environment as well.
Thinking more on the environment, this goes back to the basics of Darwinian evolution. Darwin predicts that the fetus is determined by the environment. Several of the Galapagos experiments are observing anomalies, because they are basically restricted to live in a certain environment throughout their centuries of evolution. So my question goes to the memory of transition that explained by stability in the pathways that lead to the fetus set of mutations. Because you showed when you were showing the graph with the different, there was like one was the most stable, the highest fitness, and you should nicely show in the paper that there is one path, three paths that lead to the fitness, but there are less fit conditions that can be reached by a different path. So the environment can maybe change trajectories. And so in terms, the question was very interesting, because you could ask why certain states stay with a higher probability than you would expect by random, and that probably tells you how plastic is a species. So E. coli, the beauty of E. coli is that it's haploid, and every mutation gets expressed. There are no introns, so it's like Galileo starts with simple theory and basic organisms. But the experiments already going on Saccharomyces cerevisiae, that the next most simple organism after E. coli are also leading to the second fact that a lot of the mutations are neutral. And you ask yourself why? Because they are neutral with respect to the environment that they live in.
Now, of course, I mean, the entire issue of diversity maintained in diploid and sexual populations is sort of left out here. And in fact, I think in many, here, the view here, which comes from these very simplistic experiments, is that diversity is created by mutations. Our natural population, of course, there's so much standing variation that new mutations might not even be that important. Right? So that we have a kind of different, rather different scenario.
Other questions? Comments?
Different directions. So you, as assumed always that you have to go uphill only. Essentially, each mutation has to increase the fitness. But this gives you a huge restriction on the possible paths.
Right.
Now, how real is this? I mean, I could imagine that even if you do one step and you don't increase, but you decrease slightly, but that gives you a huge new
This is certainly true. So the second part, I restricted the discussion to this uphill path. And in some sense, I think the reason why we have done that is that it leads to a very nice mathematical problem, right? Which you can analyze, and there are all kinds of interesting properties. Now, if you look at an actual, even a simple simulation like this one, you see that the dynamical relevance of these particular uphill pathways is really limited to rather small populations. So here, and this you can see here, so you see these dashed lines here, so these are steps that actually decrease fitness. So these are steps that shouldn't really take place if you only restrict yourself to uphill trajectories, and they become quite important rather soon. So in that sense, I mean, there's a process that in this literature is called stochastic tunneling, which is exactly what you described, where you sort of generate a small population in the valley, and then from there on you can sort of go on. Of course, on a sort of coarse-grained level, you probably still have certain trajectories which in some average sense tend to go uphill, right? But this strict restriction to only uphill trajectories is a somewhat artificial setting, but it's, again, as usual, you want to have a well-defined, optimized problem, and this is why we restricted our attention to that initially.
I have a question which is maybe slightly strange for you, since I am out of this community. And I've been working for some time with a genetic algorithm for optimization. And I have never understood very well which is the number of generations which is really needed to get a good result.
Okay.
So here you're showing that you're working with kind of tens of thousands of generations. Also, at the beginning you show a graph in which there was an increase in fitness, which were stabilizing on the range of 10,000 generations. Is there a kind of general rule which can predict, which can tell something about which is the number of generations around which something good happens, or not?
No, well, I mean, so one thing, of course, this obviously always depends on the mutation rate, right? So n times u is sort of the number of trial mutations that you generate per generation, right? So that's, of course, and if n times u is small enough compared to 1, then you just have to spend a certain number of generations until something happens. I mean, this is not what you would do in a genetic algorithm, obviously, because here, you can generate as much diversity as you like. And then, of course, it will depend, I would say, it will depend on the structure of the fitness landscape, that is, how far are these good places away, right? And there has been quite an effort also in sort of trying to quantify just the speed of adaptation. So, take a simple setting like, I think, something of the kind that is also used in genetic algorithm, where you simply have a kind of fitness gradient, right? So you have a kind of uphill slope, and you ask how quickly does the population actually go up the slope given the steepness and the population size. And there, one thing that I haven't talked about here, but that becomes important, and that again is very much, of course, discussed in the genetic algorithm context, is that when the populations get large, the different mutations tend to compete with each other, which slows down the adaptation. So you cannot make use of all the mutations that appear because they are competing with each other, and then you can speed up things considerably by adding recombination, right? So by recombining individuals, you go much faster. So this speed up due to recombination is also something that has been discussed extensively in this context. But my feeling, I was recently at a conference that brought together population geneticists and people from computer science, and my feeling was that computer scientists actually know considerably more about the speed, the real runtime speed of these processes than biologists. So I think one can have probably something to learn from the other side as well.
And in biology, there is not kind of general trend which is able to predict which is the speed of of adaptation?
Well, of course, biological systems are in many ways more constrained, right? Because you have a mutation rate, you cannot just control the mutation rate. You have less control over the kinds of mutations that you generate. So I think the problems are a little bit different. But I think it's a kind of dialogue that is starting now, and I think it's quite interesting.
Maybe linking into this, if you have in nature a certain mutation rate, you can, I guess, measure that. I'm not a biologist, but I guess you can know for certain species, for certain properties, what the mutation rate is. So you can put that data in your models. If you apply this kind of modeling, where you have the right mutation rate from experimental data, do the order of magnitude of generations that you need to adapt to changing conditions, does that match what you actually see in nature? For instance, the famous, what was it, the chimneys in the UK, which became dark, and then the animals living around that, they also changed color. So it seems like in some cases where you have something happening in nature, it happens rather quickly. Let's say
Right,
not with 100,000 generations
yes.
needed.
But this comes back to your comment. I think I'm not sure how well one can quantify these things even today. But one important thing is, of course, that in such situations, one is often not dealing with de novo mutations. So you actually don't have to wait for new mutations, but you have this variability in the population, and it's sort of hidden, right? So you have individuals that already have the required genotype, but it's maybe it's recessive, so it's not really seen, and then you change the conditions, and then it can come up. So I think that of course is much faster than the processes that I've shown here. I mean, so people sometimes tell me for example, when I show this Lenski slide, in some sense, of course, this is not very impressive, right? 50,000 generations. If you translate that into 50,000 generations of humans, this is a very long time, and all that happens is that they reproduce like 50% faster. I mean, but this is of course because this is an extremely restricted setting where you don't allow a lot of things to happen. I mean, you just rely on the particular mutations that can appear from that particular initial strain. So I think therefore in nature probably many things happen much faster. But by and large, my feeling is that the field is not yet in a state where one can really, link the macro scale to the micro parameters in a very quantitative way. I think it's not. I don't think anyone. I mean, this is to me always been the big question. Looking at the biosphere, looking at 4 billion years, is it plausible that what has evolved, given what we know about population sizes? And I don't think anyone can answer that question today. I don't think it's possible. But it's of course something that one should aspire to.
Just the Lenski paper uses hypermutators in LB real growth. This is not linear localized, it's selected to the flow. It's permissive for hypermutators. But a recent study on streptomyces color shows that hypermutators do not have selected advantage over wild type unless they are present together with growth and not with wild type. So it's otherwise we would think that why not every strain is a hypermutator.
But they could also, in the long run, they could have a disadvantage, right? Because they, of course, also accumulating lots of deleterious mutations, and I think there are also models that predict that eventually they will go extinct.
Yes. And how far is the field, just one more minute, how far is the field in understanding, let's say, the role of the phenotype compared to the genotype? So the speed of adaptation of a species to changing environmental conditions, again, this kind of problem. Are all the models looking at what happens basically with the DNA and how fast that can adapt? Or has the field developed further in taking into account, let's say, what part of the genes is actually expressed in the phenotype of the species and how much that plays then a role in the speed of adaptation to changing boundary conditions?
Yeah, I mean so certainly there are people who have taken this level of complexity into account. I mean, here, so the picture that I presented here, and also that is sort of the framework of most of what we have done so far, is very simplistic in the sense that you link the genotype to fitness directly. But of course, there are all these layers in between. There's also phenotypic plasticity, so a single genotype can, of course, give rise to different phenotypes.
Is it known how important that is in natural evolution?
I think it's believed to be very important. As you know, as always, you have to somehow build up the complexity in a kind of manageable way. But we do. I mean, so for example, also another way of looking at these fitness landscapes is that they, that they come about from an intermediate phenotype, so that you have some mapping from phenotype to fitness and a mapping from genotype to phenotype. And from the nonlinearities in that mapping, for example, you get something that then looks rugged on the genotype level, and this is something that we're also pursuing.
Thank you.
Okay. There are no other questions. I think that we can thank Joachim Krug again.